First one, utility patents. For example, if anyone having the new machine, compositions, method and improvement, as well as the methodology for the particular any type of work, then he will go for the utility publication. As per my suggestion, then depending on the individual guides or depending on the individual person, it may be might be chance, it might be chances for the changes, right? Then after that, design patents. For example, if I am having the idea regarding any type of design, if I'm talking about the mobile phone design, if I'm talking about the instrument design for the particular work, so I will go for the design patent filing. Process patent, methods, process for the producing goods and services. Because if in the difference of the flowchart, I'm just differentiating between the utility publication and the process patent. Before the utility publications, you will not go for the process patent, right? Design patent is a separately work. But if you are not filing any type of work for the utility publication, you will not directly go for the process patent. It literally, for example, in the diagram, I am just showing the difference between the patents. First one is the patent publications or patent utility publication. That will be a show just a white certificate. In there the author or inventor details mentioned, full name, their affiliations, their department, their then college details, all the things with their particular topics. If the person is not filing the utility, how we can go for the patent certificate, which will be centralized in the two yellow certificate you are visualized. I'm just representing that patent certificate, or another is represented as the certificate or registration of design. So how can you just directly go for the patent certificate? So first step is you can go for the utility publication, then you refining the RQ values or RQ from the IPR, then you refile your work, then it will be a granted from it will taking time more than 1.5 year literally. Or and the last one is certificate of the registration of design, that will take time to 3 to 3 to 4 months maximum from India. So difference between the certificate, only the line mentioned the certificate, patent certificate, another one is certificate for the registration of the design. So it will be the might changeable between the we are visualizing the particular certificate of the grant. So there is a lots of chances for the confusion between the certificate, which will be a utility, which will be a process patent, which will be a design patent, which will be a utility publication. So there is a lots of parameters. So as per the discussion, I am here mentioning the crucial parameter. If anyone, apart from the scholars, apart from the delegates, if anyone wants to file the patent, so the first introduction, as I had mentioned, the filing of the design patent is called the design registrations in India, involve the several steps. It comes under the Design Act 2000 and governed by the Controller General of Patents, Design and Trademark, CDDP TM. It is a broad range or a broad department from the India, which will be governing the all the parameters of the IPR. Here I'm mentioning the process step for the filing the design patents. So if you are having the idea or if you are having the any type of design idea, right? So how can you file the patent? As a I'm a scholar, as a faculty, how anyone go for the patent filing or design patent basically. So I'm here mentioning the process step for the filing the design patent. First one is determine the eligibility. So if you are filing the patent or this work is already granted from the IPR, you just modified as per your knowledge or as per your skill, or you can just say that this is my work. So I'm the filing the design patent. So in the IPR session, there are the lots of the chances from the rejections of the particular patents, right? So after the rejections, literally I'm just saying after the rejection, there is a lots of the chances for the imprisonment as well as the fine from the government of India that your work is already filed from the IPR. So how can you file as per your that you are mentioning this is your work, right? So first one is determine the eligibility, the originality, basically the originality or nobility of your work.
System achieve targeted drug delivery system because nanoparticles target those site of action where disease start, where disease initiate. That's why start the targeted drug delivery system, reduce immunogenicity and improve the drug safety and therapeutic efficacy of drugs. No, when conventional or traditional medicines is available in market, then why start ingredients? Actually, the conventional route of administration is lesser patient complaints, high entration and drug level in body, more chances of side effect, and more frequency in dose administration. But when we talk about the NDDS is the better patient complaints, minimal alteration in drug level in body, reduced chances of side effect, lesser frequency of dose administration. Because this system helps to deliver the right amount of drug for the right length of time in the right place in the body. That's why I start the targeted drug delivery system. Drug delivery system is basically of two types: controlled drug delivery system and target drug delivery system. Start controlled drug delivery system is an ideal controlled drug delivery system is the one which delivers the predetermined rate locally or systemically for a specified period of time. Novel drug delivery system start the controlled drug delivery system because in controlled drug delivery system, repeated administration to overcome non-compliance, but also helps to increase the therapeutic value by reducing toxicity and increase the bioavailability. Here is the plasma drug concentration profile. In plasma drug concentration profile, the graph between time and plasma drug level. Here mentioned the MAC and MSC level. MAC is the minimum effective concentration and MSC is the maximum safe concentration. When our drug is below MAC, then it is called subtherapeutic level. For example, when we need about 650 mg paracetamol tablet for 25 years old lady or 25 years old man, anyone but when we do not take about 650 mg paracetamol tablet, and in this case we take only 325 mg paracetamol tablet, then it goes into the subtherapeutic level because it does not cross the maximum dose concentration. That's why this is not controlled for the sustained release action. But in the sustained release, prolonged release, extended release tablet, it goes into the Cmax level. This is called conventional tablet capsules or conventional dosage form, emulsion, suspension, tablet, capsule, or any other dosage form, for example, semi-solid dosage form. Here is the mentioned toxic level. Actually, the toxic level, when the drug goes into the maximum concentration after cross the Cmax, then it goes into the toxic level. Toxic level or toxicity, it shows the higher level of bioavailability. That's why this is called toxic level. But here to mention the subtherapeutic level, sustained release manner and controlled release manner. When we start the metformin tablet, metoprolol tablet or any other PPI, omeprazoles, these all are under sustained release. Sustained release or controlled release, that means the 24-hour the drug will be maintained our therapeutic concentration and it release slowly, one by one. For example, here is the control release. These are the matrix form. And here, one by one drug will be set under the polymer matrix. When you start first drug release, then you start second drug release, then you start third drug release. This is the polymer manner of the drug. This is called, that's why it is called the novel technique, the advanced technique here. First, in the conventional or traditional medicines, will be taken 4 times in a day. This is more patient complaints. But when you start the NDDS, it implies a predictable reproducibility in drug release kinetics. Because sustained drug delivery is that type of system which achieve a prolonged therapeutic effect by continuously releasing medications over an extended period of time after administration of a single dose or only a one dose. Only one dose it take for the achieve medication for extended release tablet. But when we talk about the nanoparticles, start the polymers. Polymers actually the monomers, combinations of monomers will be formed for the polymers. Polymers are very large molecules made when hundreds of monomers can be joined for long chain. The word polymers come from the Greek word poly means many and mer means the parts. In these pictures, start the polymer. Actually, polymer is the type of excipient. We all are know that API is the active pharmaceutical ingredient. It shows the activation, pharmacological effect, or therapeutic effect. Polymer is the type of excipient. It only support for the medicines for release one by one. Nothing else. Polymer is not the API. Polymer does not show any type of therapeutic effect, any type of pharmacological effect. Only API, only drug will be show any types of pharmacological effect. Polymer is the support for the drug by releasing one by one. Next, start the targeted drug delivery system. First is the controlled drug delivery system. Second part is the targeted drug delivery system. Controlled drug delivery system further discuss about the means controlled manner, predetermined manner. But second start the targeted drug delivery system is the sometimes called smart drug delivery system. Is a method medication to a patient in a manner that increase the concentration of the some parts of the body relative to others by route of implant. For example, this is nanoparticle protecting coating. The protecting coating is the polymer coating because the polymer coating